It's the top button there. There you go.
The start. Good. So most of the work I'm going to talk about is I just, as you mentioned before, I just started my final tasks. Before I was working with Biophysical Storage at Princeton, and in my second year of ICAM fellowship, I worked with Mark Erickson and Sue Coppersmith at UW Madison. So I'm going to talk about work done at Princeton and Madison. And basically, we started a project, and the goal was to measure ligand-induced conformational changes in proteins, which I immobilized on both surfaces. And this has a sort of a double relevance here. First of all, it's, as we all know, the function of protein is related to shape, and the conformational change being transition between different shapes. So from a fundamental point of view, you would like to know different conformational change, how they are related to biological function of the protein. And second, this could lead to a fabrication of protein-based biosensors. Since proteins are so specific in grabbing different ligands, then once we understand these fundamentals, then we can go and pattern protein sensors for different analytes. Now, when we started this, there were a lot of challenges. So one of I'm not going to talk about today, but this was very important, was how you make so many proteins, because you need quantities in millimolars. And for anybody working with on protein purification, that's a major challenge. That was sort of solved by, partly by luck. There was somebody at Princeton at that time, David Wood, who's now at Ohio State, that developed a very elegant method for purifying proteins using what is called inkings. I'm not going to talk about that, but I'll be happy to discuss after. It's a very elegant method, and it doesn't use chromatography, which most biologists use, and increases the yield about 1,000-fold. So that was very important. Our project would not have worked without this new method of expression purification. So what I'm going to talk about today is how we do protein on surfaces and how we detect the interaction of proteins with different ligands. So, well, we use the atomic force microscope, and the advantages of the atomic force microscope, it's a general platform, has a high spatial resolution. It's very good, obviously, in measuring and controlling the magnitude of the applied forces and the direction of the applied forces. Moreover, if you immobilize proteins on surfaces using the atomic force microscope, well, you have a well-defined location where you can put the proteins, and I'm going to show you how to do that. You can image the proteins and measure their conformational change. It could be change in height, or I'm going to show change in friction, of course. And everything, very important, is done in the buffer solution. So there's no change. It's fluid cell at the FM, so you don't expose the proteins even to air, or obviously you don't cool them. So the proteins remain in their native state for most of the experiments. So we choose one protein that was well studied. It's the maltose binding protein, and basically it consists of crystallographic structure. It's well known. It's two domains, and there's a binding pocket in the middle. So upon binding maltose, you have this transition between the open state and the closed state. And if the protein is oriented like this, then you have a change in height about 0.7 nanometers from going between the open and closed state. Okay, so that was well known. The problem is how to put this protein on surface, and to do that, we used the technique of nanografting, which was developed by Gang Liu here at UC Davis. She did that before coming here, but she's now here as a professor of chemistry, and we collaborated with her through ICAM. And first we start with the gold surface, and this is covered by a self-assembly monolayer of a protein-resistant molecule. I mean, the protein does not absorb to these molecules. It could be, for example, an alkyl tile with a polyethylene glycol on top. And so you need a tile to fix this molecule to gold, so it forms a self-assembly monolayer. Then with the atomic force microscope, you scan that at low forces, choose the region of interest where you want to put the proteins, and just by increasing the force applied to the FM tip, you induce a chemical exchange reaction between the proteins which are in solution and the substrate. So you remove basically the alkyl tiles, and one by one the protein will self-assemble. So that's directly self-assembled. And at the end of the day, then the end of the experiment, you measure the height of the protein, the patch of the protein you make above the self-assembly monolayer, and that's going to show you if the protein is the correct orientation. So for example, well, so I'm going to show you the data, but Well, like I mentioned, you want to put the protein in the substrate, but how do they stick there? Well, one way to make them stick is to insert the cysteine linker that has a sulfur, so a tile group that will bind to gold. So that's what we did by engineering here the C-terminus. So there's a linker that was inserted there, and the proteins are in this orientation, so they have the binding pocket toward the solution. Okay. And so if you do that, then here is an example of a patch of proteins. You see the height above the self-assembly monomer is about 1 nanometer, and then knowing the height of the self-assembly monomer, you have the height of the patch, which matches pretty well with the data from crystallography. So confirms the protein orientation. This is for many patches, and you see it's a narrow, actually, because it's a zoom in here, but it's a narrow range of heights, so they pretty much all of them are oriented, I mean, they are in parallel to each other, right? So now, well, having the protein here is the question, okay, you have the protein on substrate, but maybe there is some sort of rearrangement of the amino acid, the protein, the binding pocket, might change, and the function might not be preserved. So to check that, we measure the frictional force between the FM tip and the protein patch for different maltose concentrations. So the ligand in this case is maltose. It binds to maltose binding protein, obviously. And for different concentration, you measure the frictional force. So here is, this is the same scale, but there's the patch here. It's very low concentration, and then the friction is increasing above the patch. It's also increasing above the substrate. So if you normalize that, you actually, so the normalized frictional force versus maltose concentration gives you a binding isotherm. And from that, you can extract the binding constant, which is pretty in the range of what's observed in solution for this protein. So indeed, the protein is active in the substrate. So that's the proof. By getting a binding isotherm, to show that the protein is still active. And moreover, if you want to build a sensor now, you just need to measure the frictional force, and you just extract the concentration. Okay, now this was one example. Another thing you can do with this is just to, having the proteins on the substrate, you can apply a force with the FM tip and see what's happening. Maybe you can induce this conformational change between the open and closed state. So indeed, by measuring, so here's the change in height of the protein as a function of the applied force. Obviously, height is above the substrate is decreasing with increasing force. So this is two set of data points. One is for the maltose binding protein with no maltose, and the red points are maltose binding protein with 1 millimolar maltose, which all the proteins are in. If you remember the binding isotherm, that's where all the proteins are in the bound state. So there is a difference in those two curves, and then if you look at the mechanical work performed by the tip, it's just the area here in the curve, force versus height, and you can calculate what is the difference in mechanical work. So is this the area between the two curves here, and if you divide by the number of proteins which are compressed by the tip, it's actually getting close to the transition energy between the open and closed state for this protein. So then we came up with a simple model. And again, I'm not going to discuss that because I want to show what I did at Madison with other systems. But the thing is, the model here is very simple for this protein. You have the cysteine linker, the two domains I mentioned at the beginning, and in solution, it's like they are connected by a spring. Of course, the spring is not there, but these are hydrophobic interaction. So when maltose binds to this, it's just inducing, so this is the open state, then maltose binding induces a transition between open to closed. So there's a rearrangement of the amino acids, and the protein goes to this state. There's a certain free energy change between this and this state. Both are stable states. And we have actually used this conformational change using the FM. That was the data I showed you before. And we have a proof for that. But since we talked about living matter, and we heard about viruses, which are not quite living, some people, proteins are not, we can debate a long time if they are living or not, but nobody would argue that neurons are living systems. So now, basically, this is work I did at UW Madison, and this was collaboration with Mark Erickson, Sue Coppersmith, and people from neuroscience, well, anatomy and biomedical engineering, and also Gang Liu here at Davis. And, well, the idea here is we want to study how neural neurons grow on substrates. And this actually is a sort of a deep question in neuroscience, because there are very few cases in which you can study the interaction between neurons in well-defined conditions. So you can, of course, in the developing brain, you can study how the neurons grow, and they respond actually to topographical and actually molecular cues. But developing brain is a very complex thing, and we're talking about complexity, right? And you have millions of locally, millions of neurons and thousands of connections. It's very hard to decipher which, and thousands of possible, different types of proteins there that direct the axonal growth. So you want to study this in a well-defined condition. And the way we choose to study this well is to locate neurons on substrates. And actually, this again the ICAM patterning I showed you before, to do protein patterns, and then the neurons will follow those protein patterns. And then we can actually decide where we want the neurons to, the axon to find a dendrite, or two axons to form a junction. And so we have the influence of chemical and geometrical factors in a controlled environment. So actually, technically, the technique by which we put proteins in this case on substrate is slightly different. It's called nanoshading. But anyway, so basically here it's not directed cell assembly, just because the area is much larger. You just remove this, and then the protein will stick there in all possible orientation. So if you notice here the scale, these are two examples of protein patches, and this is polylysine, so different type of protein. It's 10 microns before I show you a few hundred nanometers. But here are squares that you can make 10 by 10 microns using this technique, and then you have squares and lines. And I'm going to use both of these patterns. So what's happening is that, so these squares of proteins are surrounded by a sea of a molecule which is also resists like cell absorption, and polyethylene glycol is a good example here too. So the neurons won't stick here, won't adhere here, but they will adhere to the protein patterns. And so if you do that, and the neurons are really far away, by far away I mean 100 microns or so, these are individual cells. Okay, they stick there and they die. After 5 days, they don't, meaning that they don't grow any processes. However, if you now provide patterns, and if you make, so there are squares and lines now that guide the growth of axons and dendrites, so that you can then make a connection between, so synapses. So these are cells, two pair of cells, PDL lines, so polyethylene, polylysine. So you can decide that they will grow and will connect. And you can do the same in laminin, a different type of proteins, okay? So one direction this leads to is to fabricate actually patterns, so real neuronal network if you want on a chip. And so here's an example of an L shape. So you have 10 cells, they connect. Okay, sometimes you have cells that adhere to the substrate positions where, here there's no square intended, but you have defects. But you see they die actually, they're really far away from, as I showed you before, from typical scales of 100 microns. So if they don't see a neuron around, they can move their processes on this polyethylene glycol substrate. These connections are spontaneous? They are induced by drawing those protein lines. So we make these sort of patterns where the cells will stick here, but then you make lines, okay, and they will grow processes along those lines. And depending on how long the line is, you can decide where they go. So it's a way to control where they go. But they are spontaneous in the sense that once you make the line, they just grow. Yeah. So the same, so again, you can use different types of proteins, and actually that's an interesting direction. Now, for example, some proteins, axon responds better to some proteins and dendrites to other proteins. So there's a fusion protein, F1, you'll see that axon, it will show that axon differentially grow on that one, whereas dendrites don't. So now you can actually distinguish between axon and dendrite by patterning different types of proteins in the same substrate. So that's one direction. Another direction is actually to understand how neurons communicate with each other. So that's actually a hard problem. Do they live longer if they start talking? Yes. Meaning that, so the way actually this is done is 5 minutes. So we make the patterns, and then we culture the neurons and keep everything for 5 days. And then we look at the result. Now there is a machine you can do real time using the transparent stage of your instrument, but we didn't have that at that time. But living longer, yes, up to 5 days, they're alive and fine. Whereas the others, which don't have the connection between the die. Within a day, you said? We have within 48 hours of their. Yeah. So So now, so basically the other direction is, so you can go and make now, patterns of neurons in the substrate. The other way, you can actually understand something about how the neurons would grow processes when you have like in isolated conditions, so let's say 2 or 3 or a few neurons, right? So for example, one thing we show is that they do follow the protein. So these are the protein patterns. So the cells will stick to the squares, and then this one grows a process along those lines, and then actually turns 90 degrees. Whereas here there are no lines, and you see there are some processes growing, but it's actually not. Although it's seeing the other neuron there, but it's not growing across the peg. Whereas this neuron actually grows the process and makes a right turn. So this is a way if you want now, if you have another neuron, you make a connection here, but not with the other one. So actually how this happens is not well understood. And the thing is, we started now even simpler systems. So for example, just make these squares and look at now increase the distance between the squares. When there's 2 squares, so just squares, no lines in between them. So you have 2 cells, they appear here, they start growing processes. It is 25 microns or so, or less. They grow a lot of processes even across the repulsive peg. So they don't care about the protein. They see each other, they communicate, they send neurotransmitters, they will make connections. If you increase this distance by about 50 microns, well, they do grow some processes, but you can see they don't make connections, actually fewer processes. And if you increase this even further, you see less and less processes and shorter. And actually, we map this sort of quantitatively. You can see, so the length of the process is a function of the distance between the pairs is exponentially decreasing. And the number of processes as a function of distance is decreasing more or less linearly. Okay? So we try to understand that using a several component diffusion model. And because how they actually communicate and what's released and what's how do they make the actual connections is not understood in this, even for simple systems, right? In the same type of thing. I'm not talking about in the brain you have hundreds of different types of neurons, and they sort of find each other. And this signal is not, it's, the details of that is not completely not deciphered yet. So just to finish, I guess, so you can obviously now make lines. So the same type of experiment, but now they do grow lines. So here you can see, for example, they connect across the repulsive peg over 70 microns. So once you give them a direction to grow, so before it was about 35 microns that they make connection, then they don't connect anymore. But here, once you provide them a direction of growing, they even grow across this repulsive substrate over 70 microns gap. So the little bit here is the gap between the lines. And of course, if you increase it further, then they stop, so they don't go. So, okay, so this is the sort of the results, and come to my conclusion basically. So I talked about nanographing, way to put proteins on substrates. So one way to use this technique is to study proteins in well-defined locations and look at transition energies and conformational changes. The other way is, once you have the proteins there, well, put some cells on them and look at how the cells grow and interact with each other. That was, thank you.
Yeah, great, thank you. Questions for Cristian? Are you going to continue this work on neuronal growth?
Yes, absolutely. This one because, well, first of all, you have to have the right tools. So actually,
yes, right.
Madison, as I said, the instrument we had, the coming first microscope, was good for nanographing. However, it did not have the transparent stage, so you cannot see what's happening real time. So yes, I'm going to get final optics with that.
And so are the studies all directed at the issue of growth, or will you also be interested in sort of studying assemblies of neurons using this technique?
Well, you can do both. So first of all, I'd say the first step is to understand how do they actually grow on these protein patterns and why do they... there are definite differences between when you make a pattern and when you don't make a pattern, and what type of protein directs certain growth. Right. So once you understand that, yes, you can use that now and make more complicated circuits, neuronal circuits on the substrate. And now the idea would be to combine these microfluidic channels. Actually, I did some work on that too, but I had time. Right. So now you can put everything in microfluidic channels and supply the right nutrients, and actually you can make the neurons fire, so you can see the real-time communication. By inducing neurotransmitters.
But by using the proteins, you get a much, I mean, there are people like Eshel and Jacob who have built large sort of self-assembled cultured neuron networks, but with the use of the proteins, you're saying you can also sort of have more control over how you assemble the networks, which is pretty nice.
Yes, and having control on how you assemble them, you have control where. The problem is when they make a synapse, right?
Yes.
If they're just a few neurons, it's very hard to measure at that level what's happening. But if you have a way to control that synapse, then you pre-pattern electrodes, or even you can use the FM tip as a movable electrode and look at changes in electrostatical potential, for example, as they. So that's the direction, actually.
That's nice.
Understand those.
That's nice. Any other questions or comments for Cristian? Okay, well, thank you very much, Cristian. And next up is Justin Burton, who is at